your flowering, reproduction by sexual type in the flowering plants. You have seen the different parts of the plant like the stamen, style, stigma, petals function, and also the sepals function. Now let's talk about the pre-fertilization structures and events. So before the fertilization is occurring, we have many events that are taking place, like the formation of your flower, the formation of your stamens, formation of your spore mother cells, also called as meiocytes. So formation of meiocytes is a very important process, because without the formation of meiocytes, maybe without the formation of male or the female structures, the pre-fertilization structures, if they are absent, the final fertilization can never occur. What are the structures which are involved in the pre-fertilization event? They are the stamen, microsporophylls and sporangia. So today here we are going to study in detail about the male structures. The male structures. So first is the stamen. So what is stamen having? As I said, anther, which is the uppermost part, and then it is having a filament which is exposing the anther to the outer surface. Stamen or microsporophyll, also called as male sex organ. So in the higher plants, what is happening? The male and the female structures are completely different. The male structure has the pollen covering over it, and also it is produced from the microsporophyll. Why we are saying micro? As their structure is smaller than the female gamete. We call the female gamete as macro, whereas micro is the male. As it is smaller than the female sporophyll or gamete, it is given such name. So stamen or the male sex organ can be directly present over one flower or separately on two different flowers. It consists of two parts. The uppermost part, as you see, you can just pluck a flower in your home, and when you observe, it is having two parts. The uppermost, which is thick and bilobed, means it is having a depression in between it. This upper part, which is the anther, is having a depression in between it, and it is dividing the entire anther into two parts. So it is called as bilobed in few flowers, and it is having a filament or the long tube like structure which is exposing them, so that the pollination from the male to the female structure is becoming easy. This is the anther, and the lower tube like structure is called the filament. Anther is a broad knob. Why we are saying broad? It is more in length. It is more in length compared to its width. When you see the anther, it appears somewhat like this over the filament. So as it is more in its length compared to its width, it is called a broad knob. Knob, you know, when you use to open and close your doors. Like fertile structure. It is a fertile structure. Why? Here the number of cell divisions are taking place, and meiosis is also occurring, where the chromosomes are divided into half. A typical angiospermic anther has two lobes or two divisions in it, and such anther is called as dithecous. Di means two, thecous means anther or the male structure. So as it is having two depressions, it is called as dithecous. When you see here, this is the anther. You can also observe a depression here, a small depression here, which is dividing the anther into two parts. Next you have the filament. When you cut it down in the cross-sectional area and you observe it under the microscope, you can see. So there are two lobes. Why it is appearing as four lobes, but why I am saying two? Because these two lobes are having interconnection, and these two lobes are having interconnection. But in between, whatever space is there, that is having connective tissue in it. It It is having the connective tissue in it, completely separating both the elements. So inside this lobes there is the formation of pollen from microspore mother cells. So from the microspore mother cells, the meiotic division is occurring, that is reduction division. The number of chromosomes are becoming half, and that half of the chromosomes whichever are there are two of them are being put in one pollen grain. Two of the cells. After one division, after every one meiosis division, 4 gametes are formed. 4 gametes are formed. And in this 4 gametes, 2 gametes of each are put in one pollen grain. So 2 of the gametes are put in one pollen grain. So finally when you see, the pollen grains are created inside this depression which is called as pollen sac. Pollen sac is holding the pollen grains, and when they are having or when they reach the time of maturity, these pollen grains are released. And this is the line of dehiscence. Dehiscence itself means bursting out. Dehiscence means bursting out. So these are the lines of dehiscence which on complete, when this pollen sac is completely filled with pollen grains, this line of dehiscence or this area gets opened up and they break out so that the pollen grains move out and go towards your female gamete. And this is the stalk over which the anther is staying. This stalk is also called as filament. Coming to the female here, this is the entire female structure. As I said, the female structure is called as carpel, or it is also called as pistil and gynoecium. Anything that is representing the same, which is extending up towards the style, which is the tube-like structure, and finally stigma, which is this stigma is having the capability to take up the pollen grains which are of the same type or the same genetic material containing plants. So once these species of the same plants, so once the stigma of the species of same plants is innervated by the same pollen, this pollen will come and fall over the stigma, and only that which is compatible with the stigma has the ability to germinate. So once it germinates, it goes towards your carpel region. Here then anther, that is the uppermost bulged out area. Then the filament is present, which is the long tube-like structure. The filament and anther together called as stamen. When you take exactly the macroscopic structure of your anther, you cut it down, you can see they appear as 4 lobes. 4 lobes, and again these 4 lobes are interconnected in between by a connective tissue. The material which is present in between is a connective tissue which is holding the 4 lobes together. These 4 lobes are having what? Microsporangium in it. Microsporangium. Sporangium, microsporangium is having microspore mother cells in it, which are the cells which have the ability to undergo meiosis to give rise to the gametes which are having half the number of chromosomes, which are called as microspores. And in this picture you can also see tapetum. Tapetum is a layer inside the anther which is protecting the pollen grains or protecting the spore mother cells. Then you have the filament. A diathecous anther consists of 4 microsporangia, 2 in each lobes. When I say this is a, if I draw a picture of a diathecous anther cross section, so it appears like this. These 2 are the 2 lobes, but it is having 4 microsporangia. So each one of it is a microsporangia. So 2 diathecous, di means 2. So 2 lobes with 4 microsporangia in it. 2 2 in each lobe. So 2 microsporangia, 2 spore mother cell containing regions are present in one lobe. Example of it is Datura. You may have seen Datura plant. When you take cross section, you will see this 4 lobes in it. This is how Datura will appear outside. When you take cross section, you are observing these are the 2 lobes. These 2 lobes are interconnected by a connective tissue, and all these 4 lobes are having microspores in it. So microspores are nothing but pollen grains. The other name for pollen grains is microspores or the male gametes, whereas the pollen sac, this entire, so these black dot like structures are the microspores or pollen grains, and this dark brown structure is called as microsporangium. Dark brown structure is microsporangium, also called as pollen sac, which is having microspore mother cells in it, which divide or undergo mitotic division. One microspore mother cells will give rise to 4 microspores. This is the animated structure. You can see this is the pedicel, sepals, then this is the filament along with anther. Monothecus means only single lobule, consists of 2 microsporangia in one lobe like hibiscus. You can see when I take, this is the hibiscus plant, and if you take one part of this anther and divide it into 2 halves, you will observe there is only, and you should remember this examples hibiscus, datura and all, because they will ask you questions based on numericals on this topic. So 2 microsporangia are present in monothecus plant. Monothecus means one single lobule is present. Example is hibiscus. The main speciality of this hibiscus plant is whatever the filaments are there, these all filaments are of different length. The filaments are of different length. So once they are of different length and they are condensed or they are packed together, they appear as a single unit, but they are of different lengths. Filament. So as I said, till now we have read about anther, the uppermost elongated and enlarged part which is having the ability to divide by the meiosis. Next is filament. It is a sterile. Sterile again means filament, whatever cells are present in this filament are not undergoing meiosis, or they are not undergoing meiosis or reduction division. The chromosomes in this sterile part are not dividing into half. As the chromosomes are not dividing into half, it is the sterile. It is not directly involved in the fusion of the gametes. They are long and slender stalk. Stalk means a tube-like structure. And the proximal end of the filament is attached to thalamus and petal or tepal. So it is very important. They'll ask you, it was asked many times in examination, where is the proximal end or the lowest end of this thalamus of this filament attached to? So it can, if I say this is the thalamus, over this you have the gynoecium, then you have the androecium, petals and sepals. So this filament part is attached to the thalamus or the base. So, or it can be attached to the petal, as petals are just adjacent to, in some plants as petals are just adjacent to the filament, petals are slowly fused with the filament, or they may be tepal. Tepal means thalamus plus petal. So stamen plus the petals, fusion of the stamens or the filament part of the stamens with the petals is called as tepal type of arrangement of your filaments. Here you can observe the different lengths and also the anther size. Each one, this orange color tube like structure is called as filament. Here the pink is small and the large structure is the filament.